Hi everyone, today we are going to speak about the advanced structure of the antibody. So without wasting much time, let's just dive into it. The structure you can see on the screen is called an immunoglobulin. It's also called the antibody, and this is also called the Y-shaped structure. Now, it consists of 4 protein chains, as you can see on the screen, 2 identical light chains and 2 identical heavy chains, and they all are bounded to each other using disulphide bond, which is also known as the SS link. And this structure, this shape, helps it to bind to the antigen and also to trigger the immune response. Now, the light chain and the heavy chains. So each light chain consists of 1 variable light chain and 1 constant light chain, as you can see in the pink color here. And each heavy chain consists of 1 variable element and 3 of the constant elements. So variable means that this specific region change itself, and the constant region just doesn't change in shape, size, or its chemistry. And one last important thing about the heavy chain is that the composition of this heavy chain determines whether it's going to be a IgG or IgA or IgM class of immunoglobulin. Okay, so variable region and the antigen binding. So variable region is right at the tip of the antibody, and it consists of a variable light chain and a 1 variable heavy chain. And this can change its size and shape according to the antibody, according to the size and shape of the antibody. So as you can see on the diagram, this antigen binding site and the other antigen binding site on the same antibody, they are identical, but this won't be identical in any other antibody if that's not the same antibody. One important thing here is that the constant region just doesn't change. That's why it's called constant region. So the region which is connected by the SS link or the disulphide bond is known as the hinge region, and it is very important for the flexibility of the antibody. As you can see that it will allow some movement in the antibody if the antigen is a little far away or the shape of the antigen is not very constant, because it's not the whole antigen which will bind to the antibody. It's certain regions in the antigen which are recognized by the tip of the antibody, which is the antigen binding site. So it is important that there must be some flexibility, because one region could be on this side which the antigen can bind, and there could be another region a little far, so it will be able to stretch and bind itself. Now, constant region and the main parts of the constant region are the CH2 and CH3 as displayed here. This is also known as the Fc region or the effector region. This is what binds to the immune cells. For example, NK cells, which are also known as natural killer cells, macrophages. It also helps to activate the complement system, and it also helps in something called opsonization, which means that many antibodies can bind to one single pathogen or antigen, and then this will be the area which will go and get itself attached to a Fc receptor on one of the immune cells, and then it will be engulfed by the immune cell. Next are the carbohydrate groups on the Fc region. Now, the importance of this carbohydrate group is it provides stability, it provides and complete the 3D structure of a protein, which also helps them to go and bind to the Fc region, and it also helps them in other sort of immune interactions with other cells. Thank you so much everyone for watching. Please like and subscribe, and this video is going to be part of a bigger playlist on immunology crash course, so please stay tuned for more videos like this. Thank you.